My name is Dr. Stephen Barr, and I'm an associate professor at Western University. Today I'm going to give you a preliminary summary of some of the data we have obtained in collaboration with Aero HygenX, testing their UVC light-emitting array machine against SARS-CoV-2 and its variants. Now, before we get into the data, I'll just give you a little brief background to this virus and its consequence on human health. So about early 2020, a virus called SARS-CoV-2, which is a coronavirus, it emerged from the animal population and was exposed to the human population in a setting that allowed this virus to be transmitted between humans. And due to the infectiousness of this virus was able to survive in these human hosts, replicate, and spread with rapid pace. This has resulted in a worldwide pandemic, as everyone knows. There's over 191 million people infected with this virus, and millions, over 4 million people have died from this virus and its disease. So why is this virus so dangerous? Well, if we take a look at the biology of the virus, this is shown here in just a little schematic. The most important part of this virus is the outside. And the outside of this virus contains the protein binding components that will allow it to recognize and bind to a human cell. And if it can bind and recognize a human cell, it has the ability to enter that cell, replicate, and make more copies of itself. That's basically the whole purpose of a virus, is to replicate and make more copies. And so the virus, when it has acquired this ability, it becomes very dangerous because it can survive in its host for a period of time that will allow it to be transmitted to another human host. Now, typically, humans and other hosts have very strong immune systems that have evolved over millions of years. And typically, these immune systems can recognize and attack this virus before it has time to replicate to the point of causing disease. Now, sometimes when the virus is unknown to the human population, it hasn't had time to learn about this virus and develop a protection or a defense. And as a result, we are at the mercy of this virus and how fast it can replicate in the body and as a result cause disease. Typically, when these new viruses emerge from the animal populations, it hits the population, the human population, very hard and quick, often resulting in immediate death in a lot of those immediate infected hosts. You can take a look at a lot of the epidemics of Ebola when these outbreaks occur. They're usually fast and furious, depending on the virus, of course. Now, coronaviruses have learned to adapt to its host quite readily. And so what happens is that it can get into that host, replicate, find ways to evade our immune system long enough that it can survive and replicate. Now, when this virus emerged early 2020, labs across the world raced together, including ours, to develop a vaccine against this virus. And typically what is done is that initial virus isolate that emerged in, for example, Wuhan, China, would have been studied by these scientists, and they would identify a component of that virus, which ends up being that spike protein found on the surface of the virus. Now, they would take that protein and turn it into a vaccine. So the reason for this is that this vaccine containing that outer protein would then be used as a delivery mechanism to vaccinate people. Their bodies would take this vaccine, learn that outer protein, that spike protein, learn to recognize it, so that when the real virus then comes and infects that person, we've already been exposed to that spike protein. We know how to recognize and attack it. And then what would happen is that when that real virus comes in to infect the person, in a matter of 3 or 4 days, we would be on top of it. We'd elicit this very strong, robust immune response and end up killing the virus. So this is what was initiated, early in 2020 after that virus emerged. So labs around the world started developing a vaccine against that Wuhan isolate, and in particular using that spike protein. Now, when this was occurring, other things were happening with the virus as well. So as everyone knows, it took about a year to develop this vaccine, test it, and distribute it to the population. So during that year-long period, the virus has been changing. So it's a smart virus. Viruses are very smart. They mutate a lot. And the reason they mutate is to really avoid or evade that interaction with the human host. So this could be a vaccine, it could be a antiviral protein that attacks viruses in the body. Whatever that interaction is against that defense system, the virus is trying to get away from. And what it does is it mutates certain little sequences in its genome so that the virus itself can change ever so slightly. And if it changes that ever so slight amino acid change, for example, it would change the look of that virus just a little bit. And that change is enough to prevent that interaction with a vaccine or an antibody or some other antiviral protein in the human host so that it can't interact and attack that virus. Then what happens is that virus can replicate, survive, and as a result, it will end up causing disease in the host. Okay, so our body is really trying to maintain this interaction by developing this immune response, developing neutralizing antibodies that will bind to the virus, kill it, and keep the host healthy. So this sort of battle that goes on, it goes on all the time when a person is infected with a virus, and usually our immune systems are strong enough that we can overcome this and clear the infection. Okay, and depending on how fast we can do that can also drive how fast this virus can spread between people and, as we know, across the globe. So our bodies really did not have a good leg up on this recognition step, allowing the virus to survive and cause disease, leading to a worldwide pandemic. Now, these mutants that arise from this evolution of the virus are called variants. So they are basically the same virus, but with subtle changes in its makeup, the way it looks. And this change often leads to an increase in transmissibility. It could lead to more severe disease and hospitalization. It can lead to a stronger defense against our defenses. So, for example, vaccines would be a little bit less effective, for example, or it may just, prevent detection by our diagnostic procedures. Okay, so these subtle changes is what gives rise to variants, and if they can carry out a lot of these characteristics, they are then defined as a variant of concern, typically by the WHO. And so these variants of concerns, as we all know, the alpha, beta, gamma, and now the delta, which is starting to emerge around the world, are variants of concern. So what about these initial vaccines that we were working on about a year ago? Are they going to be effective against variants? So the question is, or the answer is, yes. Now, if we take a look at the protective effects of these vaccines against that original Wuhan isolate, we can see that all the three vaccines that have been distributed, developed, for example, the Pfizer, the Moderna, and the AstraZeneca, they are all very effective at preventing symptoms from COVID-19 based on infection by that Wuhan isolate. Now, for example, are these vaccines effective against the Delta variant? The answer is yes, but to a less extent. Okay, so they're not as effective, but still effective enough to prevent symptoms. Okay. Now, when we take a look at the emergence of variants, we get concerned because we always wonder, well, are our vaccines going to be effective? Are we going to have to go back to the drawing board and develop and test and distribute new vaccines against variants? That's a concern that still remains today as these variants are emerging and evolving. But when these variants emerge, they emerge very quickly. And that first variant that emerged was the alpha variant from the UK. So towards the end of 2020, you can see here that the UK variant started to predominate among all of the infections worldwide. So that Wuhan isolate virus started to disappear and be taken over by this new variant. And what do these variations, or how do these variations cause these new transmissions? Well, if you take a look at, for example, the UK variant, the alpha variant here, the B117, you can see that a lot of the changes or the mutations that occur in a very important region of the virus, and that is in that spike protein. So that's that protein on the outside of the virus that our bodies recognize first, and that's the basis of all the vaccines. So clearly, if these things start changing, our vaccines may be not as effective because it's not looking like that original spike protein. And so as these mutations occur, it can enhance the infection of these viruses. So for example, a mutation in that spike protein will cause it to change its structure just a little bit. That allows it to get into cells more efficiently, faster, and because of that, it can replicate better. Okay, and there's other mutations that occur that also confer a selective advantage, a survival advantage to the virus. For example, some of these mutations may allow it to evade our antibody response. Okay, our antibodies are what recognize the virus and lead to its destruction. So if you mutate those particular regions that recognize these antibodies, the antibodies will no longer recognize it, and the virus would survive longer. And the major mutations that occur, and it doesn't have to be a lot of them. For example, in the UK variant, there was one mutation in the spike protein that really made this virus more contagious. So that one mutation can occur at any time, and it happens quite rapidly, especially when it's forced to change. So if we're trying to control this virus, it's going to do everything it can to get away from that. And if it means mutating one thing here, it still survives. That's a success for the virus, and it goes on and survives. So if you take a look at some of these other variants that are out there, the UK variant, for example, the Alpha, the Brazil, and the South African variants, they will all have similar mutations that would allow its survival advantage. Now, as we are experiencing right now, there's additional variants such as the Delta variant that is emerging, and others. In the US, there's a lot that are starting to pop up here and there. But in general, these variants are starting to contain combinations of these mutations. So whereas one variant such as the UK will have a very important mutation that allows it to survive, the Delta, for example, may have that same mutation plus another one, or plus another one, to really give it even more of an advantage to survive. So that's where things become concerning and driving the need for additional methods and technologies to control the virus. At some point, a variant of concern may become a variant of extreme importance where we have to redesign our vaccine. Probably 6 months. So let's period of time, 6 months, as we're learning, right? We're trying to control, we're putting in other health measures, masks, sanitation, but there is a need for other technologies, and that's where Aero HygenX Ray machine comes in, and it is able to provide that missing link, or provide or give us that time to really develop new vaccines or new technologies, or to get on top of things and try to help prevent these worldwide shutdowns and lockdowns. So the technology that Ray has invoked is the UVC light emitting technology, and it's a technology that is not unknown to researchers. We've used UV light to inactivate a variety of different pathogens and proteins for our own various studies. And when you look at what happens with the UV light on these viruses and pathogens, what happens is that UV light will penetrate into the pathogen and cause a number of things to occur that's damaging to that pathogen. One of those is the cross-linking of DNA. So the genetic blueprints of these pathogens are very important for replication and to make more copies of itself. And what happens is UV light will cause mutations in that genetic blueprint, which will ultimately prevent that pathogen from replicating. Other consequences of UV light exposure is that proteins will become cross-linked. So the UV light will actually cause this sort of cross-linking effect, which basically glues proteins together. And when they're glued together, they can't function the way they're supposed to. They're stuck. They form this big sticky mess that basically leads to the destruction of that pathogen because it can't replicate. And what happens is the cells will eventually clear up all those dead pathogens and maintain our health. So what we at Western have done is we have a containment level 3 facility in the impact facility of Western. And what this allows us to do is it provides a safe environment that's under negative pressure. We get into these big suits that have our own air and allows us to study different technologies and different pathogens and find ways to kill these pathogens, to study them, how they replicate, to find weaknesses. And so when Aero HygenX approached us to take a look at SARS-CoV-2 using their UV light emitting ray, it was a perfect opportunity to jump on this. And so what we did was we set up these experiments in the lab where we have concentrated solutions of virus. So we have the original Wuhan isolate virus. We have a variety of different SARS-CoV-2 variant viruses. And what we do is we take these concentrated virus droplets, so the solution, the liquid that contains very high copy numbers of these viruses. And what we do is we put them on, for example, in this case, a hard surface, a hard plastic surface. We allow these viruses in the solution to dry. And this dried virus is then propped up in an apparatus that will be exposed to the UVC light from ray. And it does this at a 90-degree angle. We keep this at about 1.5 meters away, which is a typical distance in a, for example, the airplane cabin is about 1.5 meters on each side. So that ray, when it's going down, it emits UVC light and it will hit our test subjects here at 1.5 meters in distance. And so what we do is we set up different time exposures. We did a variety of different times to see how long it would take to kill the virus. And we would measure the light emitted as well at that same time and at the same point. And what we do is after exposure to ray, we would then take these dried virus samples. We would add liquid back to these dried viruses so that we can remove that virus from the surface. Once we remove it from the surface, then we are able to put them into another dish with healthy cells. And what happens is that any virus that's left in that virus droplet that we took would infect cells and kill them. So if we see dead cells, we know that there's virus around. And we have ways to determine exactly how much virus is in that sample. And that is what we did for each of these different viruses after exposure to the different times of the ray. And what we found was quite striking. And we tested time intervals from about 2 seconds all the way up to about 120 seconds at 1.5 meters away. And as you can see here with the U and T samples, the U is the untreated, whereas the T is the samples that were treated with ray. And as you can see here in the untreated samples, we have tons of virus still viable after that drying and removing process. We have more than 5 logs of virus, infectious virus. However, in stark contrast, when we treat these samples with ray for as little as 2 seconds, we are unable to recover any infectious virus from that sample. So we're seeing about a greater than 5 log reduction in viable virus. So from this was originally on the Wuhan isolate. We found that ray is extremely potent at killing any viable virus. So of course we now have taken this a little further, and this is just an example of here what it looks like under the microscope. If we have cells that are uninfected. You have these nice round, healthy looking spots on the dish. After infection, you can see that the cells that are kind of black and rounded up and in the process of dying, these are all infected cells. And if you take a look at the 2-second exposed virus samples with Ray, you can see that there aren't any detectable infection here, and it resembles the uninfected sample. So it's very striking, a very potent effect. We've now begun to take a look at the different variants and seeing how well Ray can kill these variant viruses as well. Now, as I mentioned, these viruses are still, these protein, they look exactly like the Wuhan isolate. They have subtle differences in structure. And what we can see here in preliminary data, we've done a 10-second exposure. We can see that easily after 10 seconds, we see no detectable virus from either the beta variant, which is that South African variant, or the gamma variant, which is the one that emerged in Brazil. And so from both of these, we're seeing a greater than 5 log for the beta and about greater than a 4.5 for the Brazil. So we're repeating this more, and we're going to add additional variants as well. But it seems like the killing potential of Ray is quite potent, and I would be surprised to see if it did not have a strong effect against the Delta variant as well.